FIHI is a project involving the use of hyperspectral imaging for the inspection of food products. FIHI in fact means food inspection using hyperspectral imaging. And in this project we are investigating specifically the use of this special imaging equipment called the hyperspectral imager in order to inspect the presence of contaminants in food products.
As part of the food production chain, bacteria and funguses in food products must be tested, and any food products that contain those must be eliminated from the food chain. As a part of this testing, certain food samples have to be destroyed, and these tests can take up to 7 days in some cases, or are expensive to the point where certain companies cannot even afford them.
There have been various situations where there were contaminants that led to people getting ill because of undetected pathogens that were present in food products. The idea here is to be able to detect those pathogens by carrying out basic, just taking images of food products and being able to identify the presence of these unwanted contaminants before the food reaches the supermarket or ends up on somebody's table.
The technology, if produced by the FIHI project, is aimed at providing a non-contact, non-destructive system to detect these microbes and allowing for even smaller companies to have this testing implemented within their production lines.
A hyperspectral camera in concept works similarly to a standard camera, which is typically referred to as an RGB camera. So we have a spectrum. So when we refer to a spectrum, if you look at a rainbow, there are the colors of the visible range of the spectrum, but there are also other wavelengths which we don't see with our naked eye that extend beyond those colors that we typically see in the rainbow. And with an RGB camera, so with a standard camera, we typically get information about 3 bands, red, green and blue. Those are combined together to give us one single color image, which gives us all the visible colors we typically see. With the hyperspectral camera, what changes is that instead of having 3 broad bands, the R, G and B bands, we have several narrow bands next to each other that extend beyond the typical visible spectrum. So that way, with a hyperspectral imager, we can get much more detailed information about something we are imaging. So rather than just kind of 3 values, these RGB values, we can get hundreds of values associated with different parts of the spectrum that also extend beyond what we can typically see. And this can give us much richer information about an object we are looking at than if we had to stick to a normal RGB camera. So after we take a picture of an object with a hyperspectral imager, we end up with what is known as a data cube. So as I was mentioning, for a normal camera, you actually get 3 images, the R, G and B image, that are combined to obtain one single color image. With a hyperspectral camera, we have these hundreds of different images that can be considered as 2 dimensions in space, as is normal for an image, and then you have these hundreds of layers for different wavelengths. So that's why it's called a cube, because there are kind of 3 dimensions. So we end up with this data cube, from which we can extract the information on different frequencies, different locations of the target image.
The concept behind the FIHI project allows for the development of a system that involves a camera and a computer, which can easily be deployed in many different factory type of settings, and even can be used as a portable system that allows for taking pictures of food samples and processing them, and then based on those pictures you can say whether a food sample is contaminated or not. Moreover, in most cases in the food supply chain, we would maybe test every 100th sample or every 50th or something like that. However, with the hyperspectral camera, we can test every single item, as it's not a destructive technique and it's a non-contact technique.
This project involves a collaboration between the Department of Food Science and Nutrition the Centre for Biomedical Cybernetics, both within the University of Malta, as well as FarmFresh Limited. The project is funded by the Malta Council for Science and Technology, specifically by the Fusion R&I Technology Development Programme. The next step for this project is to do further testing in industry, to test further on food products. Up to now we have mainly focused on cheeselets, ġbejniet, but the idea is to investigate the utility of what we are developing on other products as well, and hopefully in the near future we'll be able to deploy this technology in industry.
The hyperspectral camera at the moment has been optimized for detecting microbial contaminants. However, it can be redeployed for different purposes, such as testing for these other contaminants that may be present in food samples. As part of the future of this product, of course, the hyperspectral camera has many different applications, such as testing not just contaminants which are microbial in their nature, but also metals or antibiotics or any other chemicals that might end up in the food supply chain.